Welcome to EduDose. Today, we uncover a hidden danger in pharmaceuticals, pyrogens, and how the LAL test helps detect them. Plus, a real-world case study that shows why this test is a lifesaver. Pyrogens are fever-causing substances. The most common are endotoxins from gram-negative bacteria. They can sneak into pharmaceutical products through contaminated water, glassware, or containers. Even sterile products can still contain pyrogens, and that's dangerous. In the body, pyrogens trigger fever, chills, and in severe cases, septic shock. To remove pyrogens, we use distillation, ultrafiltration, or dry heat at 250 degrees Celsius for 30 minutes. Traditionally, pyrogens were detected using the rabbit Pyrogen test, slow, costly, and involving live animals. Today, the gold standard is the Limulus Amebocyte Lysate test, the LAL test. It uses blood cells from the horseshoe crab. When endotoxins are present, they trigger a clotting reaction. Advantages? Sensitivity down to 0.01 EU/ml, faster results, and no live animal testing. Let's see why this matters. In 2018, an IV fluid manufacturer recalled several batches after hospitals reported fevers in patients. The fluids were sterile, but the water system had biofilm from gram-negative bacteria. Sterilization killed the bacteria, but not their endotoxins. They used outdated pyrogen testing and missed the contamination. If they had used LAL testing, the problem could have been caught before distribution. The recall cost millions, but worse, it risked patient safety. Lesson learned: pyrogen testing is a non-negotiable shield for patient safety and product quality. To recap, pyrogens are dangerous. The LAL test is quick, sensitive, and essential in pharmaceutical quality control. Stay curious, stay learning. This is EduDose by Dr. Satish Polshettiwar.
If you found this video helpful, please subscribe, like, and share it with others who'd benefit.